I want to start out with thanks. First of all, to the organizers, of course, for involving me in this astonishingly great symposium. Thanks so much for the previous speakers who were just thrilling to listen to and have given me renewed hope that we're going to be able to really do something with respect to fixing some of these horrible conditions of the brain, as well as just understand the glories of the nervous system, which is quite remarkable. I also want to thank all of the students and postdoctoral fellows who've worked on this project that I'm going to be talking with you about today in their own native languages. And I've just had the extreme good fortune of having remarkable students and postdocs and colleagues to work with. And I feel very, very fortunate about that. And I wanted to show you their native languages in thanking them, because I think it's really an important thing for students to think about. We heard about a collaboration in Europe on the brain project, but really science is such an extraordinary international discipline, and scientists collaborate across just incredible distances, incredible cultural divides, and it's really for the students out there, I really need to tell you, it's an extraordinary profession from that standpoint. So I work on protein folding. Whoops. What happened to my... Oh, there we go. I work on protein folding, and the essence of the problem is that in order for proteins to function, they have to get into these very, very, very complicated specific shapes. They come out of the ribosome as a long string of amino acids. They fold into these very specific shapes. And if they don't get those folds exactly right, they won't function. And even worse, in many instances, if they don't get their folds right, they can go off and do very, very bad things. One of the things they can go off and do is cause cancer, and another thing that they can do is to create neurodegenerative diseases. And the reason why this is such an extremely difficult problem is that the environment in which these proteins are folding up and finding their proper folds and achieving their proper functions is, from the standpoint of biochemistry, almost kind of ridiculous. It's extraordinarily crowded in there. And the proteins are, it's not just crowded. There's an extraordinary amount of kinetic energy in the system. Proteins are bumping into each other and crashing into each other all the time. And actually, it turns out this kinetic energy and crowding of the system is actually what makes life possible. It wouldn't be possible without that kind of crowding. It makes all kinds of reactions happen that otherwise wouldn't happen. But it really does create a big problem in terms of protein folding. And so these collisions that you're seeing here are from a simulation by Adrian Elcock. But the important thing to think about is that that simulation, which represented a few seconds in real time, actually didn't represent a few seconds realistically. It actually represented only a few 1/millionths of a second. So when you start multiplying those collisions and those things happening on the scale of a neuron having to live for 100 years, you can imagine why there are so many problems that related to protein folding. So there are lots of ways in which we came to an understanding of this, and there are many different people involved from many, many different angles came to realize that this protein folding problem is indeed a real problem. Here's one way that I got into it, and that was by doing something called heat shock experiments. And basically what you see here is in very different organisms. These are yeast cells, these are Arabidopsis seedlings, and these are human tumor cells. In these very, very different organisms, if you give a mild pre-treatment and then expose the organisms to high temperatures and lots of other stresses as well, that pre-treatment will provide a tremendous amount of protection, and they will die if they don't get that pre-treatment. And as you can see, this is just extraordinarily highly conserved. It's true of every organism that we know of on the planet. Does this thing where they can respond to mild stresses by getting ready to withstand much more severe stresses. And what they're doing during that time is making new proteins. So what we have here is a simple way for the students. We radioactively label the proteins with amino acid precursors, and then we spread the proteins out according to their molecular weight, just so we can see changes in what the cells are doing. So this is what the kinds of proteins the cells are making at normal temperature. And then during that conditioning treatment where they're getting ready, they're changing what they're making. They're making new proteins. And it turns out as the same response is produced in virtually every organism that's been looked in. In fact, I don't know of an exception. And there's some slight differences between the responses of vastly different organisms, but the general principle and many of the actual individual proteins are conserved across all of biology. And they not only are induced by many stresses, they provide protection against many different kinds of stresses. And so this is a very, very broadly used survival response. It's very deeply rooted in biological systems. And so when you think about that, it's not terribly surprising that it affects our biology and human medicine in many, many profound different ways. And I just want to mention 3 different ways that we're looking at it now. My work always used to be really very basic aspects of protein folding biology and biochemistry. But increasingly, as we have come to know more and more, I think we really can start to approach some of these really dreadful disease situations. And so, for example, we're working on infectious diseases. When organisms come into our body, they undergo a big change in temperature. That's a protein folding stress. And moreover, these heat shock proteins help them to evolve drug resistance, and many other aspects of infectious disease are tied up with these protein folding responses. And they play a big, big, big role in cancer biology. We've found that this response is turned on in helping the cancer cells to survive in every cancer that we've looked at, just about. And in terms of neurodegenerative disease, well, here's where we're really between a rock and a hard place. What's happening is that cancer cells are actually taking advantage of this stress response. They're using our own stress response, survival response, to grow and evolve and to kill us. And then our brains are not using their survival response in any effective way to prevent these diseases, all of which are the major neurodegenerative diseases are all protein folding diseases. And so we think, actually, and one of the things we're attempting to do is actually block the use of the heat shock response in both the tumor cells and in stroma, and that might prevent cells from taking advantage of the survival response. And because cancer treatments are usually short-term, we think that this might be a viable strategy. I don't think it's going to be likely to be a very viable strategy to turn on a heat shock response in neurodegenerative diseases, because we'll have to treat people for years and years and years. And if we're upregulating the heat shock response in general, it's the survival response that I just told you helps to fuel the malignant state. I think it would be just too dangerous. So what we're doing instead with respect to neurodegenerative diseases is taking a more individualistic approach to try to find things that combat the initiating protein misfoldings that are part of the onslaught of changes that drive neurodegenerative disease. And so if you look at the brain and think about the proteins that are involved in these various diseases, here's a protein called TDP43, and you see big blobs of misfolded, gunky protein in the brains of people with frontal temporal dementia and people with amyotrophic lateral sclerosis. Different cells, same protein misfolding and causing disease. Same thing with respect to alpha-synuclein. Alpha-synuclein is very strongly involved in Parkinson's disease. It's also involved with dementia with Lewy bodies and other diseases. And it's misfolding in different places. There's Alzheimer's disease. Anyway, all of these diseases are characterized by gunky globs of misfolded protein. But we don't actually think any longer that it's the big globs of the misfolded protein that are the problem. It's the early misfolding intermediates that seem to be the really toxic stuff. And so this is really a big problem, as you've heard already today. As we're pushing back our age of death from so many, many other causes, we wind up, I think, on the road to ruin here because this is a highway leading to disaster, and from neurodegeneration, there is no exit. we do not have a means of treating neurodegenerative diseases, although the last talk gave me hope that at least we could be able to push it back with some fairly effective strategies. So we got this idea just because we've been working on this protein folding problem for a long time, and we know how highly conserved it is. We thought, well, maybe we could use a really much more simple organism like a yeast cell to start studying some of these processes and what's going wrong, and try to find something that might fix it. And the general reaction to this line of reasoning was, are you crazy? Really, it was quite an extremely strong reaction against our doing this. And I can understand that, actually, because thinking about the talks you heard today, this extraordinary thing called our brain is the most unbelievable, highly connected circuit where cells are communicating. The average neuron is making connections with 1,000 other neurons in the brain, each one individual neuron on average. So how could you possibly model this in an individual single cell? Well, the reason we think so when looking at the initiating pathologies is because yeast has such highly conserved biology, and because it really is a highly sophisticated eukaryote. It's a simple little microorganism, but it's much, much more complex than bacterial cells, and much, much more like our cells. So all these different compartments that you can see here, all these different cellular structures that are really vitally important for the function of our neurons are also vitally important for the function of these yeast cells. And there is no organism on this planet that we can manipulate like we can yeast cells. And this is thanks to some brewers who wanted to make much better beer about 150 years ago, actually starting with the Carlsberg brewery. They actually realized that you could get genetic variants that held true to type, for example, and that would make beer of one type or another. And since then, this organism has been studied unbelievably intently in many different applications. They make alcohol for us. They make a lot of other products for us. They're really. I have had a dog that I really loved, but I think yeast is mankind's best friend. Really, is just amazing what they do for us, and they have a remarkably robust high-throughput screening platform. They can create that, and I'll show you how we do it. And the other thing is that the basic pathways that are perturbed in those very diseases that I just showed you actually turn out to be perturbed by those same proteins when we put them into yeast. And those include things like the protein folding and quality control system I was just telling you about, agents that help proteins to get folded in the first place, keep them folded, prevent them from doing bad things when they do misfold. Those are all very highly conserved. Then there's a lot of lipid biology is actually very highly conserved. Vesicular trafficking and fusion, and I'm sure you all know that the Nobel Prize this year was given to Randy Schekman and Tom Sudoff for elaborating how vesicle trafficking is used to communicate between neurons, and that was really co-equally discovered in yeast and in neurons, both making tremendous contributions to that. Lysosomal and peroxisomal function, autophagy, a mechanism by which cells eat the bad parts of them that are going wrong. Programmed cell death, very complicated regulatory cell cycle. Mitochondria, the energy factories of the cell that empower yeast cells just as well as they empower our cells. And as I mentioned, they have this unrivaled toolkit. And oddly enough, I actually think the fact that they're single cells is an advantage in this particular case, where we want to start looking at the initiating, precipitating pathologies. And the reason for that is because having done a lot of experiments now with neurons in a dish, it is very, very difficult. And one of the difficulties is that you don't just culture the neurons, you culture the neurons on beds of glia. And the neurons make connections with each other, and they're talking to each other, and they're talking to the glia, and the glia are talking to the neurons. And there's a complexity in terms of when you start to see pathology developing there that's really, I think, quite difficult to untangle. So using these single cells as a starting point on occasion, we think can be very helpful. Now, you might think, well, how are you going to ever find anything that's going to make a difference in neurodegenerative diseases? And I wanted to say not only are the biological systems conserved and the genes that drive and regulate those biological systems conserved, but when you look at major blockbuster drugs treating human beings, the statins are the #1 most prescribed drug ever. Lipitor, simvastatin, those act by influencing cholesterol synthesis, by hitting HMG-CoA reductase, and when we want to inhibit HMG-CoA reductase in our yeast cells, we give them Lipitor. These very highly conserved proteins, you can find compounds that actually affect them. So there's reason to hope, at least, that this might work. So what we've done is we've created different yeast cell models that are expressing those different proteins I showed you a little while ago. And what we do is we put them under the control of a galactose-regulated promoter. So yeast cells love sugars, and that's why we love yeast, because they turn sugars into alcohol. But they regulate sugar metabolism with incredible efficiency. And so in the presence of glucose, those genes are going to be completely off. The cells aren't going to be making anything. But when we put them onto galactose, they turn on the expression of the proteins we tell them to make, and it creates toxicities. So this is alpha-synuclein, the protein I mentioned was involved in several different neurodegenerative diseases. And you can see that we've created strains that have expressed different levels of the protein. And I want to mention, you'll see that the protein is very bright because it's on a bright GFP, but we're actually not making all that much protein. We're not making much more protein than neurons are making per total cell protein. In fact, we're making, if anything, a little less. And we've created models of cells that are making A beta, which is a peptide involved in Alzheimer's. We are TDP43, which is ALS, Huntington, FUS3. And as you can see, I'm only showing the high toxicity strain here to match up these different models. And I just want to emphasize that these different models have very equivalent levels of toxicity. And so it might have been that this would provide a way of just finding non-specific things that affect protein aggregation and toxicity. And I would have held that, well, even that's worth doing. But it turns out that that's not the case. The things that we see are, for these different proteins, when they cause toxicity, are completely different. We see different cell pathologies, there are different genes that rescue them, and there are different compounds that rescue them, although there's some overlap, hopefully, with some of the compounds. So this is just one example of a biological response that's characteristic of the cells that are expressing alpha-synuclein. This is using an antibody that detects reactive nitrogen species that are damaging a lot of proteins in the cell, very specific proteins, but a massive storm of reactive nitrogen species. And we're only seeing that response in the cells that are making alpha-synuclein, even though they're all equivalent. And I'm going to come back to this response a little bit. So I'm going to tell you primarily about alpha-synuclein. We've taken that one the farthest. We started with it. It's one of the first we started with. And a reason for deciding to try alpha-synuclein first was that this is a very simple little protein. It's just a simple lipid-binding protein. It doesn't have all kinds of complicated domains. It's 14 kilodaltons, small. And it has some very basic biophysical properties that are believed to be involved in the disease, and that should hold true in yeast, we thought. So, for example, when the protein is not associated with membranes, and it's in a natively unfolded state, it has a lot of random coil structure and conformational dynamics. When it encounters a phospholipid membrane in the cell, it becomes structured. And so it's peripherally associated with that membrane, and so it's free to come on and off of that membrane quite easily, actually. And when it does come off, it gets prone to forming a variety of different misfolded, what we call oligomeric states. And so this protein, importantly, is heavily involved in Parkinson's disease. It's seeing those aggregates of this protein, this misfolded globs of that protein, is characteristic of Parkinson's disease. But as I mentioned, it's true for different cell types in different neurodegenerative diseases. So we thought, well, there's some basic aspects of misconduct that this this protein is doing that can play out in different cell types. Maybe they play out in a yeast cell. So here we are. We switch alpha-synuclein on and off with galactose. We hook it up to GFP, and we've tested it without the GFP too. But we like to do many of our experiments with GFP so we can see where the protein is. This is simply a fluorescent protein that it will shine green back at you when you shine blue light on it. And so if we express 1 or 2 copies of that protein in the cell, what we see is that it goes to the plasma membrane. And that was nice because we know that in the brain, it hasn't been known what alpha-synuclein was doing, what its normal function was, but it's associated with the little vesicles that Andy was talking about earlier today that are involved in moving neurotransmitters around the cell. They move a lot of things around in the yeast cell too. And if it was trafficking, one of the reasons it's called synuclein is because it's found at the ends of the nerve cells. And so if it was trafficking in a similar way in a yeast, we'd expect to see it at the plasma membrane. And that's what we see. And then when we express just a little bit more of it, things start to look pretty bad. And if we express twice as much, things look much worse. And these cells grow fine. Those cells grow slowly, intermediate toxicity. And these cells just plain die. They start dying after about 8 to 10 hours. The reason why that was really intriguing and exciting was that replicates the extreme dosage sensitivity you see in human disease. So this is one of the very few human diseases that you can get simply by having an extra copy of the wild type gene. And if you have 2 extra copies of the wild type gene, you will get a much earlier onset and much more virulent form of the disease. So, and that's unusual. There aren't that many proteins that show that kind of dosage sensitivity. So this is where we screen every gene in the genome for things that will affect the toxicity. And this is where we screen for compounds for rescue because, as I'll show you, lots and lots of things are going bad here. So what do I mean by screening? Well, it's really a lot like panning for gold. You go through an awful lot of sand, boring sand that's not doing anything much and is not giving you much, and then all of a sudden, you start to get some gold. And that's basically what screening is. We screened 6,000 yeast genes. That's the entire yeast genome. Many of those genes are very closely related to human genes. And we found of those 6,000 genes that we tested, fully 1%, that's about 60 of those genes, affected the toxicity of alpha-synuclein. So that's complicated. That's actually unusual for a yeast screen to pick up so many genes. And what was interesting about it was that they were involved in very different functionalities. The genes that rescued, some of them seemed to be unrelated. But there was one group that was closely related to each other. Those were genes that were involved in moving those little neurotransmitter vesicles around in neurons, but also involved in yeast and moving little packages of things to the outside of the cell. So the things that moved trafficking forward rescued, things that put the brakes on trafficking made them sicker. So the natural thought was that, well, maybe alpha-synuclein is itself affecting the trafficking of those vesicles, and that's why if you the genes that rescue that would rescue the cells from alpha-synuclein. And so we simply looked. And this is electron micrographs of yeast cells, and you can see these are little vesicles that are normally trafficking to the periphery of the cell, and you start to see them accumulating even in the non-toxic state and more with toxicity. And then we asked, where was the alpha-synuclein in those cells? And we did immunoelectron microscopy, and I won't show you that, but I'll just remind you, with the immunoelectron microscopy, that's where the alpha-synuclein was. It was sitting right over the blocked vesicles. So alpha-synuclein is binding to those membranes. It's particularly causing those membranes to stick and to not move on to their next destination. It's turned out that this has now been corroborated by many studies in neurons. And for neurons, and I'll show you a few of those from our lab, but many other laboratories have corroborated it. The other way in which we look at this, electron microscopy is not a really high throughput assay. You can't assay a lot of samples that way. So a much easier way to look at problems with vesicles moving around is that when proteins are run out in the gel and you look at their molecular weights, you see them as they move from place to place in the cell, they change their molecular weight. It depends upon the protein. Some get larger, some get smaller, but things get added to them and taken away from them. And so you can tell that there's a difference in the trafficking in this cell versus this cell just by simply running the proteins out for analysis this way. So of course, all of this meant nothing initially until we were able to ask whether did the genes that we get out of that screen that altered alpha-synuclein toxicity in a yeast cell, did the homologs of those genes make a difference to alpha-synuclein toxicity in a little worm, C. elegans, which was mentioned earlier, and in rat embryonic neurons. We wanted to use two different models, one that was a wired-in nervous system and another one that was closer to home. And we tested 6 diverse genes that were quite structurally and functionally unrelated. And we tested 7 genes. 6 of them rescued the neurons from alpha-synuclein toxicity. But of course, what you really, really need to do is look at human neurons. And I, as someone who works on model organisms, I really appreciated your earlier question today about C. elegans and Drosophila. But let's face it, we have to know that it's really directly relevant to human patients. And so what we've done to do that is a variety of different things that have connected the yeast system to what's happening in neurons and allows the yeast cells to then tell us a lot more about what's happening in neurons. So for example, this is just one example. We were sitting there with this list of genes that we'd gotten out of our screen, and then all of a sudden this paper got published. Hereditary Parkinsonism with dementia is caused by mutations in this protein, which encodes the lysosomal type 5 P-type ATPase. So we looked in our yeast cells and asked if it was also going to the equivalent compartment, the lysosome of a yeast cell, and sure enough, that's where that gene goes, that's where that protein goes. And then we could use our yeast cells to try to find something out about its function. We imitated the human mutations that are associated with these genes, and we showed that they were loss of function mutations. They could not rescue the cells from alpha-synuclein. And then P-type ATPase, that indicates metal trafficking. And so we added all kinds of different metals to the cells and asked which ones were rescued by this gene. And it turned out that it rescues yeast cells from manganese. Now, the reason why that's intriguing is that manganese exposure in many different forms and ways has been associated with Parkinsonian diseases. So using yeast cell, we've connected alpha-synuclein to vesicle trafficking defects. Didn't show you, but mitochondrial dysfunction, that's that energy factory of the cell that's been connected by others to Parkinson's disease. Manganese homeostasis, iron and calcium homeostasis problems. I don't have time to tell you about that. And then to this previously unconnected human mutation in PARC9 that causes Parkinson's disease, but none of those patients had gone to autopsy and no one had any idea if they actually had aggregates of alpha-synuclein. So there's a relationship in humans when you don't have enough of that function. You get early onset Parkinson's disease. When you overexpress that function in yeast, you correct against alpha-synuclein, this major player. So what about other human variants? So what we could do was we could take advantage of vast amounts of other people's data. So there have been about, I think it's now up to about 15,000 genome-wide screens conducted in yeast. That is screening 6,000 genes against this or that or this or that. So there's this massive data of what things are genetically related. And then there's masses of mass spec data, protein-protein interaction data, immunoprecipitation data, et cetera, that connects proteins together. And so we were able to find unexpected connections between human disease alleles that are involved with Parkinson's and other related disorders, but had not been thought to be related to each other. So this work has not been published yet except for the PARC9 story. So I haven't actually named the genes here yet, I'm sorry, but it wouldn't actually be too hard to guess what they are because there are several homologs of genes that are now known to be associated with Parkinson's disease, are these yellow balls. And they are connected. These are the lines here that connect the balls are simply the yellow dots are a way of representing in an abstract way, higher order function here, representing a gene, and then there's a connection that's been made between these two on the basis of a protein-protein a genetic interaction, these by physical interactions, et cetera. And this is the sum of those really, really strong interactions. We've gotten rid of all of the ones that are minor. So that really gives us encouragement that we're really seeing things that are related to Parkinson's disease, and we can start, and other synucleinopathies, and we can start to take advantage of these yeast models. And actually what we've done recently, again unpublished, but we've started to create individual personalized medicine models in our yeast cells. So we take cells that are making a little bit of alpha-synuclein, and then we take another one of the Parkinson's disease genes and mutate that. And what we wind up is getting cells that are dying, but they have related pathologies, but there are differences between them. And I think that this is important to do because if we're going to find compounds that can maybe rescue this and be used in the clinic, we're going to want to make sure that the compound is rescuing from the same kind of pathological deficit that we are seeing in human beings. And we're going to need to stratify the patients according to specific susceptibilities. So another way that's proven to be very difficult but really worth the trouble was to use human iPS cells. So one of the people in my lab is a neurologist, and he treats patients with Parkinson's and other disorders, movement disorders. And he identified a patient that had a synuclein mutation, and we took a skin biopsy, dedifferentiated these cells with the extraordinary help of Rudolf Janish's laboratory, and then we differentiated those cells into neurons. Now, what happens is that you and I differ in an awful lot of locations in our genomes. There are many, many differences between any two individuals. So what we wanted to do was to have cells that were as close to being identical as we could get them, but that had one mutation in alpha-synuclein that would be associated with this pathology, different between them. And so we used genome editing to correct. This was a patient who had a mutation of alanine at amino acid position 53 to threonine, and we corrected it back to alanine. So we have genetically identical cells, but then we've got the problem that the cells look just absolutely fine. The patient neurons look fine, so did the corrected neurons. But what we were able to do is, and that's not a surprise, because after all these diseases strike, the people who have that mutation typically get Parkinson's at about the age of 50. But what we were able to do is, long before the cells started dying, before we saw any phenotype visible, when we actually looked for the things that were going wrong in our yeast cells and asked, are they going wrong in those patients? Can we see an early sign that that's going wrong? And we could. So here's, and they were not going wrong in our mutation-corrected lines. So this is just, I'm not going to take you through these gels. These are just, these protein bands are changing because the proteins are not moving through the vesicle trafficking machinery the way they should. Another way in which we connect these human stem cells, patient cells, is that one of the genes we found corrected, it was known, it's annotated in the genome as protecting yeast cells from nitric oxide. And yeast cells don't have their own NO synthase, so it was not thought that yeast cells made NO. But it was really of interest to us. There's one paper that came out while we're in the middle of this study that showed that they can make it under certain circumstances. So the reason why that was interesting was that in Parkinson's disease, so now we're learning from what's been known in patients, when you stain with antibodies that recognize those damaged nitrated proteins, like I showed you earlier on that blot, you see that Parkinson's patients have a lot of nitrated proteins, alpha-synuclein as well as other proteins. And so that's what caused us to go back to these models, take proteins out of them, run them on gels, and look to see if we were getting a storm of nitration. And sure enough, we got a massive storm of nitration. Moreover, that nitration was not just being released, that nitric oxide, it turns out, was not just being released all over the cell, but there were very highly focal places where that was being released, and it was closely associated with, although not completely overlapping, with the alpha-synuclein itself. And so then we went to patient neurons, and also used a sensor that would allow us to look at, here we used a chemical sensor for NO. Here we used an antibody against the nitrated proteins, and you can see that we have lots of highly focal, localized release of nitric oxide or reactive nitrogen species, and that it's corrected by the mutation correction. So now we'd like to look for compounds that might be able to rescue for two reasons. Maybe eventually they could be a therapeutic, but also they could tell us something about the biology, because chemical genetics can be quite powerful. And it's important to note that in this study here, between 1999 and 2008, there were more new chemical entities that were approved as drugs by the FDA that were found from phenotypic screens. That means a biological screen. You don't know what the compound is necessarily doing when you find it, but you find it biologically. Then there were from target-based screens. That is, you might think, oh, identify a kinase, and you want to find a compound that'll hit that kinase. But the drug companies, almost all research over that same period of time has been focused on targeted screens. That is, identify a particular protein, take it out of a cell, and in a biochemical assay, look for a compound that will alter its activity. And the reason for that is very simple. Once you have a compound that does even some magical things biologically, it's awfully, awfully difficult to figure out what the heck it's doing. And it's very difficult to mount clinical trials, especially long-term clinical trials, for example, in neurodegenerative disease, when you don't know what the compound is doing. So the idea was, might we find compounds that would fix more than one problem in the cell that would so that's why we use that high toxicity strain where all kinds of things were going wrong. Can we find compounds that will fix more than one problem? Because the idea would be maybe that compound would be hitting some really central node that radiates into different kinds of pathology. And by hitting that node, you can correct different kinds of pathology. And might we use yeast genetics, I just told you this organism is astonishing in terms of its genetics, to find the target. And so we screened 550,000 compounds. It is not currently possible to screen neurons at that level of high throughput. That's really a high throughput screen. And we did this in collaboration with two different groups, the ICCB at Harvard and the NIH. And we simply asked for compounds that would restore growth. That's all. And we've only dissected a few so far. We wanted to go in and really look at them in detail, so we took one of the earlier compounds. It's by no means the best. And we found that the compound ameliorated vesicle trafficking problems, it ameliorated mitochondria defects, it worked in the nematode, and in rat neurons, and in our human neurons too. So the compound is N-aryl benzimidazole, and I don't know how to do any chemical synthesis myself at all, but we worked with a really talented group at MIT, Steve Buchwald's group, and they made a lot of different derivatives of this to get compounds that would be more effective, et cetera, et cetera. And we're continuing to work with them on this project. But anyway, we had a high faith that this compound and very small changes you made in the compound would completely eliminate its ability to rescue. And it rescues many different signs of toxicity. So I told you that it rescues vesicle trafficking, and so you can see these big blobs of blocked vesicles. Hardly any of the cells have blocked vesicles after treatment with this compound. It restores the trafficking of proteins in the secretory pathway. It completely eliminates that storm of nitric oxide. And it also rescues patient neurons from multiple phenotypes. It restores the trafficking of group cerebrosidase. And when we look at this storm of nitration that occurs in the A53 T cells, this is the genetic correction, surgical genetic correction, and this is the correction with the compound. So how do we find the target? Genetic screens that are not possible in any other organism. So we examined every one of the 6,000 genes in the yeast genome for if we overexpress that protein, would it alter the sensitivity of the compound? We looked at 100,000 transposon mutations. We looked at deletions of just about every gene in the genome. And we looked for individual spontaneously rising point mutations in individual amino acids and proteins by sequencing, getting things that change the sensitivity of the compound and sequencing the genome. And it was really only the combination of all of these different screens that allowed us to identify how the compound was working. And that really is the awesome power of yeast genetics. It's just not possible to do all of those together in any other organism. Although certainly great strides are being made, but for now it's not possible. So we only got a small handful of genes that triangulated from all of these different screens. We tested every single one. And it turns out that what our compound is doing is activating a ubiquitin ligase. Ubiquitin ligases are proteins that regulate all kinds of protein folding and protein homeostasis pathways in cells. Lots of them. This is a cartoon of the protein structure, of the different functional domains of the protein in yeast versus in humans. As you can say, there's a lipid and calcium binding domain, there's protein-protein interactions domains, and then there's the ubiquitin ligase domain that brings ubiquitin and puts it on another protein, very specific proteins. And really only this portion has been well characterized biochemically so far. And the thing about this protein is it undergoes all kinds of conformational changes while it's doing its job. It interacts with all kinds of adapter proteins, all kinds of different substrates, and so it actually really demonstrates very vividly the power of phenotypic screens. And it also demonstrates the power of chemical genetics, because we would not have known that this protein was central importance in alpha-synuclein toxicity. Our other screens had not picked it up. But what the power of the chemical is that it could change the activity state of the protein, not just express more or express less. So I'm almost done. So it turns out that there are almost 700 ubiquitin ligases, as I mentioned, really important proteins, and drug companies have been trying to drug them, and they have not succeeded for the most part. There are very few pharmacological agents out there that affect these compounds. And I have no activators. There's actually one activator that has been characterized. So what's cool about this protein is that it functions to promote vesicle trafficking. And it does so at several of the different sites that are blocked by the vesicle trafficking events that are blocked by alpha-synuclein. And it relieves not every one of the trafficking events that's blocked, but most of them. So we have very parallel effects of alpha-synuclein on multiple pathways in the cell. And we hope that we will now be able to transition into other models. We've actually screened 550,000 compounds in our TDP43 model and our A-beta model. We've got compounds that look even better than the compound I just told you about. I think it's the power of this chemical genetic approach to find compounds which will have their biological effects, these complex biological effects that we could not have thought up. We would not have known what to do. So we've also done genome-wide genetic screens, and we're hitting all kinds of genes that have been identified as being involved in Alzheimer's disease. So we have this platform that we think helps to facilitate rapid biological understandings and identify compounds. We don't necessarily have any reason for thinking that compound is going to be useful in any of the synucleinopathies. It might turn out to be that you don't want to activate that ubiquitin ligase in those diseases, but I have the feeling that compound will be useful in some protein homeostasis disease or other. And it's just beginning. So these are the amazing, wonderful people who started the synuclein work in my lab. These are the stars of the show in terms of the recent analysis. And my wonderful collaborators are also simply a gift of God to me as far as I'm concerned. They've just been tremendously helpful. So thank you all very much.